final speaker for this session will be Hedi Young, who will be telling us about cortico-cortical loops in the mouse visual cortex.
Hi, so I'm going to be talking to you about long-range loops in the cortex and the implications these findings have for hierarchical models of the cortex. So, as you may know, the cortex is hierarchically organized with lower areas and higher areas. And given this hierarchy, we can essentially boil down long-range connections to two types. Feed-forward projections that go from lower to higher areas, and feedback projections that go in the opposite direction. Now, these different feed-forward and feedback projections terminate differently across the 6 layers of the cortex. And interestingly, these basic anatomical profiles are conserved not just across areas, but also across species, which implies that these hierarchical interactions lie at the very core of what the cortex does. Whoops. We're back. Whoop. Okay. We're good. So there are different theories of hierarchical cortical computations, and some of these theories require looped interactions between feed-forward and feedback projections, while others do not. Those that do require looped interactions include predictive coding theories and theories where feedback sends a teaching signal to train the weights of synapses, akin to the way backpropagation trains neural networks. But it would be really useful in order to be able to constrain these theories if we could find fundamental rules of connectivity for feed-forward and feedback projections. But right now, we don't know what the wiring diagram looks like. We do know that cortical areas are reciprocally connected, but that doesn't actually mean that looped computations are actually happening. For example, you might imagine a scenario whereby feedback targets cells that don't project back to it, but rather to a different cortical area. And in this case, if loops did exist, they would be indirect and involve several synapses. But if looped computations really do lie at the core of cortical function, we would expect to find direct monosynaptic loops that involve just one synapse. So we began a series of experiments to see whether feed-forward and feedback projections really do preferentially target cells that they form monosynaptic loops with. And we recorded from the 3 major classes of pyramidal neurons in the cortex: IT cells that we find in all layers that project within the cortex, and CT and PT cells in layers 5 and 6, respectively, that project outside the cortex. And we did these experiments in the mouse visual cortex, looking at feed-forward projections from V1 to higher order areas, LM and AM, and looking at feedback projections from those higher order areas to V1. And so the method we used to look at connectivity and to measure the connectivity was scRACM. In short, we inject channelrhodopsin virus to label either feed-forward or feedback projections, in this example, feedback, and we inject retrograde tracers to label cells that project to different areas. Then we take a brain slice and perform whole cell recordings in cells right next to each other that are labeled and in the same cortical layer. And during the recordings, we shine a laser around the cell to activate those feedback inputs and record the size of the response. And because we have TTX in the bath, that blocks polysynaptic activity, and so any inputs we record are monosynaptic. And so from this, we get these maps for each cell, which show both the strength and, importantly, the location of those monosynaptic inputs. And so we can just do a paired comparison and sum the total of these responses for each cell. and compare the responses and see which cell got more response from the feedback area. In this case, the cell on the x-axis, which projects to the source of the feedback, received more feedback than the neighboring cell projecting to a different area. So that's the method. If we look at the results, let's look at whether or not feed-forward and feedback projections preferentially target cells that they make the loop with versus neighboring cells that project outside the cortex. And so if we look at layer 5 and compare IT and PT cells, looking at feed-forward inputs to these cells, here's a population summary. And so each dot represents a comparison between two cells. The cell on the x-axis, as in all the plots I'm going to show you, is the cell that projects to the source of the inputs. The cells on the y-axis project to a different area. And as you can see, the cells projecting to the source of those feed-forward inputs from V1 receive twice as much input as neighboring cells that project outside the cortex. What about feedback? Well, we just do the mirror experiment, and this time we record in V1 and activate feedback inputs from LM to V1 and compare the IT and PT cell responses, and we see again that feedback preferentially targets cells that they make the loops with versus cells projecting outside the cortex. If we go down the layer to layer 6 and look at feed-forward inputs to IT cells versus CT cells, we see again that feed-forward preferentially targets cells that loop back to the source of those inputs compared to neighboring cells that project outside the cortex. And again, if we look at feedback, it's the same story. Cells that project to the source of feedback in layer 6 receive more feedback from LM than neighboring cells projecting outside the cortex. And so to sum up these two experiments, we see that in both layers 5 and layer 6, both feed-forward and feedback selectively target cells they form loops with versus cells projecting outside the cortex. All right. Okay. And so, but what about when we do the paired comparison between cells that project within the cortex? And so, really, for this experiments, these kinds of cells come in two flavors. Cells that project to the source of the inputs and neighboring cells that project to a different cortical area. And so if we start off in layer 2-3 and we look at feed-forward inputs from V1 to LM, we did not see evidence for selective targeting of cells that loop back to the source of the inputs. You can see that the cell projecting to V1 does not appear to receive, on average, more input. And the same is true for feedback. Feedback from... This thing seems to have a life of its own. Feedback from two different sources, LM and AM, we don't see a selective targeting of the cells that loop back to the source of those that loop back to the source of those inputs. And so what about if we, so basically to sum up in layer 2-3, we don't see selective targeting of the cell looping back either for feed forward or for feedback. So if we go down to layer 6, however, it's a different story. Feed forward inputs to cells in LM that loop back to V1 do receive more feed-forward inputs than neighboring cells projecting to a different cortical area. And if we look at feedback in what is becoming a familiar pattern by now, looking at two different sources of feedback in the different shades of green, feedback from LM or feedback from AM to V1, again, we see that feedback selectively targets cells they form monosynaptic loops with versus neighboring cells in layer 6 that project to a different cortical area. And so finally, in layer 5, again, we look at two different sources of feedback from LM to V1 or AM to V1. Feedback, as you can see from this reconstruction example, targets not just the perisomatic region around the soma, but also the apical dendrites. And so we did not find evidence for selective targeting of these cells when we just look at the perisomatic region. However, when we look at the apical dendrites, we saw that cells projecting to the source of the feedback inputs receive more inputs in their apical dendrites compared to neighboring cells projecting to a different cortical area. And now we'll do the mirror experiment for feed-forward inputs. So we looked at two different types of feed-forward inputs from V1 to those same areas, LM and AM. And here is a single cell example. And to our surprise, we saw that feed-forward inputs, like feedback inputs, don't just target the soma, they also target the apical dendrites. And so when we look at those feed-forward inputs to the apical dendrites, we see, again, as with feedback, that feed-forward inputs in the apical tufts preferentially target cells that make the reciprocal connection. And so to sum up then, we see that in each cortical layer, feed-forward and feedback appear to follow the same rules of connection in their projection to these classes of pyramidal neurons, despite the fact that they have different anatomical profiles. And because we look at two different types of feed-forward projections and two different types of feedback projections, it appears that this symmetry is a general organizing principle of the cortex. And what are these rules? Well, both feed-forward and feedback selectively target and form monosynaptic loops with the apical dendrites of layer 5 neurons and with the layer 6 neurons, but not with the layer 2-3 neurons. And so if we do a loop back to the beginning of the talk and come full circle to the theories outlined earlier, we do find a biological substrate for these looped computations, but only within the deep layers of the cortex. And as you'll have seen during some of the talks and posters, some of these theories implicate the apical dendrites of layer 5 neurons, and that's what we find in our data. And so I'd like to just thank my supervisor, Leopoldo, for all his help and advice, and Beatriz Belbut and Margarita Baeta, who've helped me tremendously on the project. And I'd like to thank you guys for your time. Thank you.
Time for some questions. I'll start off. So you're warning, it wasn't kind of an absolute rule. It was about a 2 or 3-fold preference. Do you think the non-loop connections, is that just kind of messiness in the wiring, or are those connections also serving some other role that's distinct from the loop connections?
Right. So as I said, there are some theories that don't require looped interactions, like theories of attention, and so perhaps those inputs, the cells that don't loop back to the source of inputs, have some role in those areas as well. So for attention, for example.
Yeah. So you think these models can kind of coexist?
Yeah, there's no way. Yeah, I mean, it's absolutely possible that these different models work in parallel.
Great. Got one over there.
Yes. So I have a question. So in these experiments, we somehow discovered a lot of loops that are have both feed forward and feedback on the same pair of neurons. My question is, how can this kind of connectivity provide the function that it can calculate the prediction error? Because we know that prediction error can be both positive or negative, and if we form this kind of loop, and are they excitatory or inhibitory connections? And if they are kind of like same for example, excitatory connections, how can I calculate both the negative and positive error with this loop?
Well, so the inputs I've been looking at today are excitatory, but we know that feedback inputs, for example, don't just target pyramidal cells, they also target interneurons close to those pyramidal cells. And some people have suggested that these positive negative predictions you're talking about might differ depending upon whether the feedback inputs are targeting excitatory versus interneurons. But we can talk more about the details afterwards. Yeah.
Thank you.
Well, let's thank Hedi and all of our speakers in this session.